Enough.
Yeah. I don't think you're sharing your screen.
Here we go. Once again, we're going to talk about the excitability of cells, right? How cells pass on signals. Well, next time we'll actually say how they pass on signals from one cell to another at the synapse. So we're going to look about electrical conduction through an individual cell, right? A couple of different kinds. And here's my haiku for this, which is there's a neurotransmitter release, graded potential, action potential, and then release again, right? So we're going to repeat this over and over again, right? It's going to be a neurotransmitter release, graded potential in the dendrites and cell body, and then an action potential down here, right? That's basically what's going to be going on within a cell right here, right? So this is starting this whole thing about neural communication, right? Neurotransmitter release at a synapse from one cell to the next. That's going to be followed by the so-called graded potential, which are going to be used for this kind of short distance within the dendrites and the cell body, which may lead to a nerve action potential, right? Which is going to be this kind of long range communication out to the periphery or from the periphery. Right? So we'll have this kind of local communication within cells. Then you'll have action potentials, long action potentials going from one region, say in the brain, all the way down to your spinal cord, a little more local communication, and then a long action potential down out to the periphery right here, right? So this, mostly we'll be talking about this graded potential and action potential and everything kind of surrounding these issues right here, right? So we'll be looking at the difference, right? We're going to be looking at this resting membrane potential, what exactly that means, and what happens when that resting membrane potential is disturbed in some way, right? And what happens at different stimuli, different places. We'll be looking also what this kind of graph actually means right there. So first, we want to talk about the so-called resting membrane potential, as well as what we're talking about there. What, when you see 0 millivolts, minus 70 millivolts, what are we talking about? So when in biology, right, you've heard like electric signals moving through you, right? We're talking about the movement of charged ions, right? Sodium, potassium, they're moving from one place to another, right? If you're talking about a wire, right, and an appliance, it's a copper wire, there's electrons moving alongside the copper atoms. In biology, we're talking about charged ions, right? Potassium, sodium, calcium, anything, chloride, any movement when we're talking about electricity. So movement from one place to another, right? A current, it's caused by a difference in electric potential. The word potential just means this difference, electrical difference between one place and another. Right? Just like when we talked about the concentration gradient, you had a lot of molecules over there, less over there, and then there was a flow of molecules. That's a little different from what we're looking at here, but there's a difference from one place to another, and that's referred to as a potential, right, or an electric potential. The way you get this potential, in our case, is that this plasma membrane is going to be an insulator, right? It's not going to allow electric charge to pass through it. It's said to provide resistance, opposition to flow of ions from one place to another. So you're separating groups of ions from each other due to this selective permeability. That is, charged ions don't go through here. Right? But there's going to be an unequal distribution of different types of ions and different charges due to the fact that this is selectively permeable. And we'll see why how you get this different distribution. So when you have this kind of difference in the outside versus the inside, you know, the cell is said to be polarized, which most actual cells in your body are going to be polarized. That is, there's going to be a difference inside the cell from outside the cell. Mostly for what we're talking about, there's going to be a difference such that we're going to call the inside of the cell minus 70 millivolts, right, on a voltmeter. Voltage, right, the electric potential, the electrical difference between these two is minus 70, right? If we consider the outside 0 millivolts. These don't really mean anything in absolute terms. It's just the difference between these two, right? If you're in a house, it's really hot outside, it's 100 degrees. Your house has an air conditioner and it's 65 degrees. The difference would be minus 35. You could say your house is minus 35 relative to the outside, right? That's kind of what this is like. We're going to set the outside at 0 and say that the inside of a cell is minus 70 millivolts. Okay? So that's the electric difference between across here, right? And then for a normal cell, when nothing else is happening right here, normal difference is the resting membrane potential. I'll use RMP quite a bit in my little blurbs there, right? Resting membrane potential. That's when no disturbances are going on. You just have this resting membrane potential of about minus 70. Right? And since this is polarized, the cell, if it goes towards 0, we're going to call that depolarized, right? So keep that in mind for a second. So resting membrane minus 70, and the minus, it's just negative relative to the outside. Again, if your house was, I could say your house is minus 35 because you got an air conditioner and 65 versus 100 degrees outside. Due to the insulation and the selective permeability, right, you got this difference in voltage, right? Minus 70 on the inside, 0 on the outside. So let me just make sure, I'm going to draw my pictures here. Let's see, here's your house. Here's a window. It's 100 degrees outside. And 65 degrees inside. That's a smile. Hey, right. So that's the difference. And you could say that the difference was minus 35 in here, right? If this was 0. If for some reason threw a rock through here, busted the window, all this hot air would come rushing in, right? Or you open the window, right? This guy over here would feel this blast of hot air over here, where it is right over here, right? You'd feel that kind of hot air rushing in, right? Because it was kept out artificially by this window. But now you've opened up that window and all this hot air is going to rush in there as soon as this permeability, as soon as this insulation thing has been removed in that case, right? So keep that in mind as well. So when we talk about the resting membrane potential, what are the factors that are going to contribute to it? We already said the plasma membrane itself, right, is an insulator. That is, charged ions don't cross. So how you get this distribution is going to be due to these kind of channels and what it lets in and what it lets out. So first off is the unequal distribution of ions, right, in the extracellular fluid and then the cytosol. The extracellular fluid, right, the interstitium around there, rich in salt, sodium chloride on the outside right here. You have a lot of sodium ions, you have a lot of chloride ions. Right? That's what's happening on the outside. On the inside of the cell, you got a lot of also positively charged potassium ions. The main negative one is going to be things like proteins and phosphate ions, things attached to phosphate ions, as we'll see in a second, right? But for this over here, these distribution of ions right here, one of the biggest, the relative impermeability to sodium, right? That is, there's not so many sodium channels which would like to go into this negatively charged area. It's relatively impermeable, right? Keeping all those sodium molecules outside, right? There are more potassium channels, right? and there's more potassium in here. Potassium would leak out when allowed, right? These are actually going to open and close kind of back and forth. They're considered leak channels. Right? But there's a difference in between sodium and potassium channels, and that'll make a difference as we'll see later on. Right? But the thing that the book, and I'm not really explaining right here, is that you got a large negative charge inside, mostly due to these phosphate molecules and proteins, right? Negative charges on certain amino acids of proteins. That accounts for most of your electronegativity of any type of cell, right? So you got a negative charge in any type of cell, even without these channels or anything. So if you have a large negative charge inside here, all those positively charged molecules want to get in. So if sodium is going to, which is a little more permeable, is going to kind of go inside the cell because of that negativity. The cell would be about minus negative 90 millivolts. But because your sodium is able to flow inward, right, that brings it to about minus 70. Okay. At some point, the sodium stops flowing in because its concentration gradient is kind of reaches equilibrium, as we say. And again, the book doesn't really describe this. That's why there's more potassium in here. It's allowed in and it's attracted to this negative charge. Sodium would like to go in if there's just less opportunity for it to do so, right? But so the fact that this is negatively charged inside, sodium would flow in. That's why you have a lot of sodium ions. It brings it up a little bit positivity from minus 90 to minus 70. I'm not going to ask you about that, but that's why they're flowing in. And then at that point, there's too many potassium ions based on concentration gradients, and then they start to flow out again once it reaches about minus 70. All right, so most anions here, big proteins and stuff, will not get through this membrane. They're stuck in here. That's why your inside is negative, okay? And then the last part of it is any minor fluctuations in it are going to be kind of kept constant by this constant movement of your sodium, potassium, ATPase pump, right? This requires energy, but it's pumping out sodium against its concentration gradient and bringing in potassium. So there's a little more sodium pumped out than the potassium brought in, right? This doesn't have a huge factor in the actual resting membrane potential actually, but it does matter in restoring it if it's been disturbed. So these are the three final factors that are keeping this resting membrane potential at around minus 70. Big negative charge by all the proteins, and sodium is kept out. All right. So what we're going to look at over here, right, is the flow of electricity around the cell. And we're going to remember the names, the parts of the names of the cell, right? Dendrites around here that are reaching out and getting signals from other neurons or whatever, or sensory receptors, as well as the cell body, right? So signals are going to flow in this direction, remember. It's going to be electrical signals coming from the dendrites toward the axon hillock, all the way down the axon into the axon terminals to the next cell, right? So this is what we're always keep this in context in your mind, because we're going to kind of get close in on a lot of these areas right here.